Allelopathy is the process where plant species interfere chemically with the germination, growth or development of other plant species has been known for 2000 years. Molish in 1937 introduced the term allelopathy and is derived from the Greek word allelon, that is of each other, and pathos meaning to suffer, which means injurious effect of one upon the other. In recent years, the term is generally accepted to cover both inhibitory and stimulatory effects of one plant on another plant. In 1996, the International Allelopathy Society defined allelopathy as follows: any process involving secondary metabolites produced by plants, microorganisms, virus and fungi that influence growth and development of agricultural and biological systems, excluding animals and including positive and negative effects, and it was given by Torres et al. 1996. Allelochemicals. Chemicals released from plants and imposing allelopathic influences are termed allelochemicals or allelochemics. Most of the allelochemicals are classified as secondary metabolites and are produced as offshoot of the primary metabolic pathways of the plant. Some allelochemicals are known to have structural functions, that is intermediates of lignification, or to play a role in the general defense against herbivores and plant pathogens. Allelochemicals can be present in several parts of plant including roots, rhizomes, leaves, stem, pollen, seed and flowers. Allelochemicals are released into the environment by root exudation, leaching from the above ground parts, and volatilization, and by decomposition of plant material. When susceptible plants are exposed to allelochemicals, germination, growth and development may be affected. The most frequently reported gross morphological effects on plants are inhibited or retarded seed germination, effects on coleoptile elongation and on radicle, shoot and root development. Next, let me move on to allelopathy as a possible cause for crop yield reduction. It has been shown that certain plants have a depressive effect on its neighbors. It is a common fact that neighboring plants will interact in a negative manner where the emergence or growth of one or both is inhibited. Several workers have shown that the adverse effect of a neighboring plant in association with others and called it as interference. The potential causes of interference include allelospoly, that is competition, the depletion of one or more resources required for growth. Allelopathy, that is the addition of chemical toxins by one or more species in association. Allelomediation, that is the selective harboring of a herbivore that might selectively feed on one species, thus leading to the advantage of another. The overall effect of one plant upon another is called interference, which includes both allelopathy and competition. The removal or diminution of a shared resource is called competition, while addition of a chemical compound to the environment through different process is called allelopathy. Some workers considered allelopathy to be part of computation, and computation has been misused by many to describe interference. In a historical overview, Wills 1985 pointed out that allelopathy is not a new concept. Theophrastus 300 BC first noticed the deleterious effect of cabbage on wine and suggested that is due to odors. A common problem in both Greek and Roman time was so called soil sickness, the declining yields of fields. They did not understood that the condition could be caused by various factors such as mineral deficiencies, toxin accumulation, pathogens, and the imbalance of microorganisms. In the 17th and 18th centuries, botanists relied strongly on a comparative approach. They compared both plant form and function, particularly in relation to nutrition. The Dutchman Beroe suggested that root exudation may play a role in plants. Stephen Ailes believed that root exudates facilitated excretion of used compounds. The theory of root excretions was a basis for the concept of allelopathy. Coming to proof of allelopathy. Many field studies implicate allelopathy, but isolation and identification of the chemical agents require rigorous laboratory effort, as shown by Putnam and Tang in 1986. It is extremely difficult to prove that any deleterious effect is due to allelopathy rather than to computation for essential products. Numerous studies have provided evidence, but seldom has a specific protocol been followed to achieve convincing proof. Putnam and Tang 1986. These authors pointed out that the shortcomings of the discipline make it hard to differentiate between allelopathy and computation. These shortcomings include a general lack of nomenclature to adequately describe the plant response that occur in this manner, a dearth of techniques to separate allelopathic interaction from computation, and a failure to prove the existence of direct, compared and indirect influences via other organisms and microenvironmental modifications. A considerable body of information has accumulated implicating allelopathy as an important form of plant interference. According to Willis 1985, Putnam and Tang 1986 and Cheng 1992, the methodology dictates certain points for allelopathic research to be established to suggest that it is operative. A pattern of inhibition of one species by another must be shown using suitable controls, describing the symptoms and quantitative growth reduction. The putative aggressor plant must produce a toxin. There must be a mode of toxin released from the plant to the environment and thus the target plant. Mode of toxin transport of accumulation in the environment must be evident. The affected plants must have some means of toxin uptake, be exposed to the chemical in sufficient quantities and time to cause damage and to notice similar symptoms. The observed pattern of inhibition should not be explained solely by physical factors or other biotic factors, especially computation. Next, we are supposed to know what are weeds are in effect. Weeds are the plants which grow where they are not wanted. They grow in the fields where they compete with crops for water, soil nutrients, light and space, and thus reduce crop yields. They also harbor insects, pests and microorganisms. Certain weeds also release into the soil the inhibitors or poisonous substances which are harmful to the plants, human beings and livestock. They increase the expenditure on labor and equipment, render harvesting difficult and reduce the quality and marketability of agricultural produce. Natural powers of weeds. Weed seeds germinate earlier, their seedlings grow faster and they flower earlier, form seeds and mature ahead of the crop they infest. Nature has bestowed these qualities on weeds so that their seeds are collected unwarily along with the produce of the crop at harvest. They have the remarkable capacity to germinate under varied conditions. Peak period of germination always takes place in certain seasons in regular succession year after year. Types of weeds. Weeds belong to the class Angiospermae, that is flowering plants, and they are divided into annuals, biennials and perennials. In addition, aquatic weeds which are submerged, immersed and floating types. Another type of weeds known as parasitic weeds, and these weeds parasitize certain host plants which they directly attack and deprive them of water, nutrients and assimilates. According to Putnam and Tang 1986, all alleged cases of allelopathy that have been studied appear to involve a complex of chemicals. No single phytotoxin was solely responsible for or produced as a result of interference by a neighboring plant. Few studies on allelopathy concentrate on the mechanisms and process involved in the production of allelochemicals. Healing 1987 and Putnam and Tang 1986 raised the question whether alleged biochemical agents were in sufficient concentration and with enough persistence in the environment to affect a neighboring or succeeding plant. These chemicals should be transformed during the course of extraction. According to Cheng 1992, allelopathic symptoms may not be manifested at the time or site where plant damage has actually occurred. Sources of allelochemicals. Radiosevich and Old 1984 stated that primary effect of allelopathy seems to result from an association with plant litter in or on the soil. Rice 1984 and Putnam 1985 reported that allelochemicals are present in the virtually all plant tissue, that is leaves, fruit, stem and roots. These allelochemicals are released by such process as volatilization, root exudation, leaching and decomposition of plant residues. According to Rice 1984 and Putnam 1985, there are 4 ways in which chemicals are released. Volatilization, that is released into the atmosphere. It is only significant under arid or semi-arid conditions. The compounds may be absorbed in vapor by surrounding plants, be absorbed from condensate in dew or may reach the soil and be taken up by the roots. Leaching through rainfall, dew or irrigation may leach the chemicals from the aerial parts of the plant that are subsequently deposited on other plants or on soil. Leaching may also occur through plant residues. Their solubility will affect their mobility in soil water. Root exudation from plant roots into the soil environment. Whether these compounds are actively exuded, leaked or arise from the dead cells slowing of the root is clearly understood at the time. Decomposition of plant residues and is difficult to determine whether toxic substances are contained in residues and simply released upon decomposition or produced instead by microorganisms utilizing the residues. Allelochemicals represent a myriad of chemical compounds from simple hydrocarbons and aliphatic acid to complex polycyclic structures. The secondary products could be classified in the following categories, but it is impossible to enumerate each and every chemical identified as an allelochemical. Whitaker and Finney, 1971, Rice 1984, Putnam and Tang 1986 divided allelochemicals into various major chemical groups. They include simple water soluble organic acids, simple unsaturated lactones, long chain fatty acids and polyacetylens, naphthoquinone, anthraquinones and complex quinones, simple phenols, benzoic acid and derivatives, cinnamic acid and derivatives, flavonoids, tannins, terpenoids and steroids, amino acids and polypeptides, alkaloids and carbohydrates, sulfides and glucosides, purines and nucleosides, coumarins, thiocyanates, lactones, octogenins. Most of the chemicals are secondary metabolites and are produced as byproducts of primary metabolic pathway. Rice 1984, Putnam Tang 1986, Rizvi et al. 1992. Secondary compounds have no physiological function essential for the maintenance of life. Aldrich 1984. Reports most frequently identified effects which are readily observed in the field are under controlled conditions. Delayed or inhibited germination and the stimulation or inhibition of root and shoot growth are often reported by Rizvi et al. 1992. The direct action involves biochemical or physiological effects of allelochemicals on various important processes of plant growth and metabolism. Processes influenced by allelochemicals involve mineral uptake, that is, allelochemicals can alter the rate at which ions are absorbed by plants. A reduction in both macro and micronutrients are encountered in the presence of phenolic acid, as shown by Rice 1974. Cytology and ultrastructure. A variety of allelochemicals have been shown to inhibit mitosis in plant roots. Rice 1974. Phytohormones and balance. The plant growth hormones indole acetic acid and gibberellins regulate cell enlargement in plants. Membranes and membrane permeability. Many biological compounds exert their action through changes in permeability of membranes. Photosynthesis. Photosynthetic inhibitors may be electron inhibitors or uncouplers, energy transfer inhibitors, electron acceptors, or a combination of the above. Respiration. Allelochemicals can stimulate or inhibit respiration, both of which can be harmful to the energy production process. Rice 1974. Protein synthesis. Studies utilizing radio-labeled C14 sugars or amino acids and traced incorporation of label into the protein found that allelochemicals inhibit protein synthesis. Conducting tissue, water relations, genetic material, etc. are also part of it. Under natural condition, the action of allelochemicals seems to revolve around a fine-tuned regulative process in which many such compounds may act together on one or more of the above processes. Allelopathic interaction between the plants have been studied in both managed and natural ecosystems. In agricultural systems, allelopathy can be part of the interference between crops and weeds and may therefore affect the economical outcome of the plant production. The search for genes involved in the production of allelopathic compounds in crop has begun. This widens the opportunity for improving the allelopathic activity of crops through traditional breeding strategies or by genetic engineering. Biotechnological transfer of allelopathic traits between species has been suggested as a possibility, and this could, for example, be from wild or cultivated plants into commercial crop cultivars. Demonstration of allelopathic activity. It has recently been said that no area of plant science has provoked as much controversy as the study of allelopathy. Generally, the difficulties of separating the chemical interference from other interference mechanisms have hindered the acceptance of many of the methods suggested to demonstrate allelopathic activity. Therefore, the validity of many test results within the field of allelopathy has been much debated. Coming to indication of allelopathy. Investigation of allelopathic activity have been initialized by field observation mainly related to changes in agricultural, horticultural or silvicultural productivity or to changes in vegetation patterns in natural habitats. Problems of growing the same crop in succeeding years because of poor establishment and stunted growth has led to investigation of possible causes including allelopathy. Allelopathy occurring among individuals of the same species is termed autotoxicity. Autotoxicity is known, for example, in medicago sativa, that is alfalfa. Biotic and abiotic factors can influence both the production of allelochemicals by the donor species, that is the species from which the allelochemicals originate, and modify the effect of an allelochemical on the receiver plant. Even though the production of allelochemicals in a plant can increase in response to stress, it is not clear whether a corresponding release of allelochemicals to the environment also occur. In general, the sensitivity of target plants to allelochemicals is affected by stress and typically it is increased. Allelopathic effect in the mulberry garden is due to the undesirable plants in the field, and if they persist for longer duration, allelochemicals accumulate in the soil, hampering the soil chemistry and soil microorganisms. Due to these reasons, quality and quantity of crops may get diminished and will have long-term bearing on the income generation of the farmers.